immune system. Specific immunity and its neighboring defense, nonspecific immunity. Three characteristics that differentiate specific from nonspecific are as follows: antigen-specific, systemic, and memory. Antigen-specific targets certain pathogens or foreign substances that ultimately trigger an immune response. Systemic allows for an active response throughout the entire body and does not limit to an initial infection site. Lastly, memory grants specific immunity with the ability to remember and fight off previous attacks on the immune system. The types of responses that involve specific immunity are humoral immunity and cellular-mediated immunity. Humoral immunity is only found in fluids such as blood and lymph. The process begins with a macrophage that engulfs the pathogen and displays its antigenic fragments on the outside of the cell. This forms an antigen-presenting cell. Holding the antigen are proteins called MHC2s, or a major histocompatibility complex, that acts as a badge holder. Next, a once naive lymphocyte called a helper T cell links to the MHC2 and becomes activated. Once activated, the helper T cell will divide and differentiate into another active helper T and memory cell. Simultaneously, as the same pathogen floats through the fluid, it is caught by a B cell's antibodies. This is the start of the antigen-antibody complex. Therefore, the B cell signals help from the newly active helper T cell. The T cell will give the B cell cytokines to allow for the B cell to divide and differentiate into memory and plasma cells. The plasma cells will then create antibodies and release them into the blood, where they will have a lifespan of 4 to 5 days. The memory cells are a key role for long-lasting immunity. In return, they generate a faster and stronger response towards any future recurring infection. This process memory cells use is called secondary immune response. When a virus invades a normal body cell, it will cause a cellular-mediated immune response. Like humoral immunity, a floating pathogen is engulfed by a macrophage, where it is digested by powerful lysosomes and other enzymes. The proteins are then shown on the outside of the macrophage, where it is now called an APC, or antigen-presenting cell. The APC cell now presents its proteins to a naive cytotoxic T cell. With a MHC1 holding the pathogen's antigen, the naive cytotoxic T links to it, resulting in its activation. Like humoral immunity and cellular-mediated immunity, helper T cells play a vital role in this process. An active helper T cell senses the cytotoxic T cell in distress and gives it cytokines. The cytokines are chemical messengers that allow for proliferation of a cell. The cytotoxic T cell will now divide into a memory cell and another active cytotoxic T cell. The memory cell is then stored for when the body is invaded again by the same pathogen. The newly activated cytotoxic T cell will target and kill any of the cells displaying non-self antigens.